And that's why, and as you'll see, in the first 40 questions, I'm just really directing you to look at certain things and don't look at the things that, if I don't tell you to look at it, don't look at it, okay? It's not that it's not important, but you only have so much time to get ready for this, and I don't want you to looking at things that or maybe confuse you, okay? All right? All right, so okay, I think I'm ready.
Yep, stay between the lines and you're in good shape.
Okay, all right, so we're ready to start. So welcome to this review. I have a studio audience here, and then some of you will be viewing this via video, and so welcome to our review. And so this is Thursday, April the 4th, and we're reviewing for the first half of the exam, or the first 40% of the exam, which takes in Unit 1, 2, and 3. And so that takes in a lot of territory. There's a lot of chapters in there. So I'm really encouraging you to focus on the things that I'm asking you to study for that part, because that's the way I'm making up the exam. So in Unit 1, Chapter 1, and let me erase this board. This was our test here. I didn't really put anything down there in your sheet that relates to Chapter 1. But there is one topic in Chapter 1 that is something that maybe maybe if you remember this, it will help you. And that's the whole idea of the genus and species, okay? Now, in that chapter, you know, we learned about the kingdom and all of that. I won't be asking any questions for the whole gamut of that, but at the end of when you go down from the kingdom and you go all the way, and the last two steps in that is genus and species. And we use that so much in microbiology. And this is the clue to you to know that you were talking about the scientific name. So this has to do with the scientific name of our organisms. And I noticed that when we did that open book test and we had open, questions, that when you were asked the name of an organism, you didn't always know to go to the scientific name. And then sometimes there was some mix-up between the name of the disease and the name of the organism. So remember, when you're reading and you see a two-part name, you're looking at a scientific name. And that stands for the genus, which is a bigger group, and then the species, which really defines what the organism is. Okay, so like we're Homo sapiens. That's human beings. Right? So we're totally, you know, in the whole world of organisms and animals, you know, that's us. And so the organisms are the same way. And so once you get to that, what we call the species level, you're looking at organisms that have that share and act in the pretty much the same way. Now we know that there are variations, just like there are variations with us, but it doesn't change the fact that we're humans. So we can, you know, have all kinds of variations, but we're still humans. We still have the same capabilities. So these organisms, so kind of think of them like that. And so when we see this, when we see Escherichia coli, and when we write it, we underline it, or if we type it, we italicize it. So when you're reading things and you see italicized print, then you know you're dealing with the scientific name. And so it's telling you that this belongs to the group genus, or genera, for more than one genus would be genera. It is Escherichia, but coli gives you the distinct the distinct organism that it is. So you have to have both of these to really be able to distinguish each organism. And some of the organisms, now that we're at the end of the course, and I know that you got this at the beginning of the course, but now that we're at the end of the course, now it's probably more important to you to understand this, or you're like, okay, now what does all that mean? So, for example, we had an organism called Neisseria, and we had one of those organisms, Meningitidis, and what did Neisseria meningitidis cause? Anybody like to answer?
Meningitis.
Meningitis. And it really was the worst form of meningitis, okay? Now notice that when they decided on the name of the organism, and people who, through history, who worked on these organisms, they get the privilege of naming the organism. So there was actually a scientist named Neisser, and so this was named, he got the organisms named after him, the genus of it. And then, so what they picked for the last name, or the species name, was the disease that it causes. So it causes meningitis, and so they just took the word meningitis and they came up with the name Meningitidis. So, but we have another one. Okay, anybody know the other organism that is also the genus Neisseria? Causes an STD.
Gonorrhea.
Yes. And so we have an organism called Neisseria Gonorrhea. Now we don't spell it like the gonorrhea, the disease gonorrhea, because the disease ends in HEA, but the organism is gonorrhea, but it's spelled OEAE. Okay, so that's the organism, and the disease is gonorrhea, HEA, and the organism is gonorrhea, Neisseria gonorrhea, but it's HOEAE. Okay, so now maybe now that you're all the way through the whole course, maybe you're starting to see a little bit of logic of why that was important to understand genus species. And you know, these organisms share, it's funny, they share some similar things. They share some of their virulence factors. And they both can attach to cells, but they just attach to cells in two completely opposite ends of the spectrum. Neisseria meningitidis comes through the oral pharynx and gains access to the spinal column and causes meningitis. Neisseria gonorrhea also attaches to the mucous membranes, but it attaches in the genital tract and causes gonorrhea. Both bad organisms, very virulent, cause lots of problems, but they're very distinct in the cells and where they're located and where they live. And the other thing that they share, is what, do you remember what their gram stain is?
Positive.
No, it's a negative. It's a gram negative. And you remember us looking at we looked at a picture of the spinal fluid, and we looked at a picture of a urethral smear, and we looked at inside the white cell, and there were these little, you remember what was inside them? No, you don't. Okay. All right, so gram negative. They're gram negative, and I used my red pen to remind you that gram negatives are red or pink. Maybe that'll help you this afternoon. I don't know, you
Yeah.
having a lab practical?
Yeah.
Okay, they're gram-negative diplococci. Okay? So basically, it's like this. They're circles. A cocci is a circle. And they are together. They travel in pairs. So two organisms travel in pairs. And so these are both this. So that was one of the things when we were doing some practicing with the names of the organisms. Generally, and I can't think of any of the exceptions, no exceptions of anything that we have studied. If they have the same genus name, if you know one gram stain, then you know the other. So they're going to be the same on that. Okay? So that's just a little bit of how to logically use that information that you're getting from the genus species name. And then there's one more thing that I will tell you about genus species is that none of the viruses are named that way. They call them groups, like we learned about the herpes group, but most of them are given the names of the diseases they cause, like rabies virus, chickenpox virus, influenza virus, and so their nomenclature is totally different from the bacteria, the protozoans, the helminths, the fungi, all those groups that we study. They will have, like the bacteria, they're going to have a genus, species name, but the viruses do not. And so technically, whenever anybody writes the name of a virus, they should not italicize it. So that's one clue, when you're reading, it's one clue for you is if it's not italicized, but it says, influenza, well, obviously it almost always says virus after it, so you know that it is a virus, but it also will not be italicized, or you don't underline it. And so the only thing we do underlining or italicizing is when we have a scientific name that is the genus and species name. Okay? So that'll lead us into chapter 2, because what I want to do in chapter 2 is give you a couple of things of chemistry to remember, and also I want to talk about prokaryotic and eukaryotic, okay? So, because that's a pretty important concept for you too. Okay, so as you see, there's not a lot in chapter 2 that I've highlighted for you. I think that the pH scale is important in that only because you're going to use it in so many different ways. We use it in micro because we always are reminding that certain organisms like certain environments, and so it's and even though chemically there's a strict definition of the pH and how it's calculated, for us it's really pretty simple for us, because the pH scale is, the center of the scale is a 7, okay? And it goes from 0 to 14, okay? And so this middle point here is a point where we say it's neutral. And what neutral means is it's not acidic, and it's not basic. Okay. If it goes to 7.01, so if it moves anywhere in the direction of 14, it's considered to be basic. Okay? So here's where we have basic, and another word that we use for basic is alkaline. Okay, so remember that is when if somebody says basic or they say alkaline, it's the exact same thing. Okay, so anything above 7 is an alkaline pH. Okay, now, oh, sorry. Little p. Okay, and then on this side, it's acidic. So if it's 6.9, it is slightly acidic. And the closer you get to 0, the more acidic something is. So these are the stronger acids down here toward like 2, 3, 4, like stomach contents would be down in here. And these are the stronger bases where it gets up towards 14. So, it's just a couple simple things that, a vinegar would be acidic, whereas Clorox is basic. So that's just, everything has, generally everything has either like a slight pH one way or the other, or a lot. So the main reason that we wanted to know that about the microorganisms is because we know that they generally don't like this part of the scale. They usually, some of them do, but most of them don't. Most of them are up around in this area. We had some exceptions that we talked about, like Helicobacter pylori that likes to live in the stomach and can survive in that acidic conditions. So that's one of the things. As you go along, I think in anatomy, you're going to learn about acid-base balance of humans, and we are slightly basic. Our blood pH is 7.35 to 7.45. We have a very, very narrow range, and if your patient gets in trouble with their respiratory system, then their pH can go one or two ways based on what their underlying problem is with that. So anyway, it's very important for us to understand that. So that's that out of that chapter 2. Also, and I'm not going to put this on the board because I want to make sure that we get to where we need to be. But in your workbook that you have, or the notes pages, be sure you go back and remind yourself as to what a carbohydrate is and what a protein, a lipid, a protein, and a nucleic acid. And so carbohydrates would be our sugars and our polysaccharides. Starches would fall into that category. And our energy is stored in the carbohydrates. And eventually, anything you take in your body that's a category of carbohydrate, eventually it's going to become glucose, because that's what you use in your blood to, fuel what goes on, all of the energy processes that you need. So that's the carbohydrates. The lipids, we learned 3 different types of lipids: the triglycerides, the phospholipids, the sterols. And so each one of them has a unique function, and that's on your notes. So go back, review the categories and what the purposes of each of those are. And then we had proteins. It's important for you to remember that proteins are chains of amino acids. And proteins are important for structure, like our muscles depend on protein. But proteins are also very important because they're the enzymes. And you remember we learned about different enzymes as we went along. The organisms have enzymes, we have enzymes. One of the ones that we studied was lysozyme, because we know it's throughout the body, like in the mucous membranes and the eye. And what it does is it will break up a bacterial cell. And so enzymes are chemicals that speed up reactions and make chemical reactions take place. So that's kind of one of the more important aspects that we used proteins, was the whole concept of enzymes. And then we had nucleic acid, and I was going to go into the nucleic acids a little bit here for you, because it came up in another chapter for us, and so I wanted to do that here to remind you about that. And so, does anybody remember what the two types of nucleic acids are? What's your chromosomes made out of? Your chromosomes.
What chapter is that in?
Chapter 2. It's the first time is chapter 2, and then it comes back in the genetics chapter.
Is that DNA and RNA?
DNA and RNA. Yay! All right. Okay, so you want to be sure that you go back and review that structure of DNA and RNA. Now, they both have a sugar molecule, and DNA is deoxyribonuclease, and RNA is just ribonuclease. Okay, so that's one difference of them. The other difference of them is something that we learned, was that it's called a DNA is called a double-stranded helix, which means it curves and it's like a ladder. It's like a twisted ladder, okay? And so it's made up, and the rungs of the ladder, it takes one chemical called a purine and one chemical called pyrimidine, and they form the rungs of the ladder, but they're arranged in a specific way, and that was one of the things that we learned. And so you have purines, and as I said, you have pyrimidines, okay, and the purines that you have would be adenine and guanine. Okay? The pyrimidines that you have is cytosine and thymine. Now this is for DNA, okay? Alrighty. Now, so but they pair up in an exact fashion, and so what you have, it was called, let me remind myself of his name, I think it's Chargaff's rules, and yeah, Chargaff's rules, and it's A always goes with T, so adenine always pairs up with thymine, and guanine always pairs up with cytosine. Okay, so they always pair like that. And it's definitely, I have that in the review games that, you know, for you to remember. And so the pyrimidine that pairs with adenine is thymine, okay? So you need to remember them all like that. Now, what happens is that down here in RNA, what you do, and if I can, I'm just going to put it above thymine, and I want you to remember that this is the RNA arrangement, and it's called uracil. So that's for RNA only, okay? So when you come down to RNA, the way it would pair up would be A and U, and then still C and G, okay? So uracil is a pyrimidine, and it takes the place of the thymine. And I worked on a way for you to remember this, okay? All right, so let's see if this helps you. I kept trying to think, okay, how am I going to remember this? All right, so if you can remember the fact that pyrimidine, okay, so purine is adenine and guanine, okay, and pyrimidine has a Y in it, and cytosine and thymine have Y's in that. So you can recognize the DNA pyrimidines by their Y, okay? Now, so what do we do with uracil? Uracil replaces thymine in RNA, okay? So what comes after T? U,
Oh, you
right?
know the alphabet.
Okay. Does that help?
Yeah, I get it.
I'm hoping that'll help. Or maybe it's just easier for you to memorize it. I don't know. So you can decide whether you like that or not.
Does it matter like how you switch those two around at the bottom, the C equaling G, cytosine equaling guanine, as opposed to the top?
It doesn't, as far as the, they do switch back and forth. But you know what, let's do that this way just so you can, let's be consistent there. Because that is one of the ways that we get variation in our DNA structure. It's like a code. So in other words, it can be AT, like the rungs can go like AT, G, GC, CG, TA, you know, so it's a code. So they can go in either order. But let's do that, and then that way, that'll also help you to keep remembering that these are purines and these are pyrimidines here, okay? All right, so that's a good point, okay? So when your notes will be consistent. Okay, any other questions about that? Okay, good. All right, so then the last thing, and this really bleeds over into chapter 4, is the whole eukaryotic, prokaryotic thing. And so let me put that up here. All right, so what I want to do is, I've got a diagram here I want to put up for you. And that's why, now you know why we use PowerPoint, right? Because it really does slow you down quite a bit. But I think this will actually help you though, really, in the long run. Okay. Oh well. I can do it. All right, so here's what we want to do, is we want to look at eukaryotic versus prokaryotic. All right, so we have I need to come on. Okay, we also have a difference because we have some organisms that don't have a cell. And so we have acellular organisms. Okay, so in this, we have the prokaryotes means that they have no nucleus. the eukaryotic organisms have a nucleus. So basically here we have two groups. We have the bacteria, which is really the only group we dealt with. And then there's some environmental organisms that are related to the bacteria called the archaea. And we really didn't study them, but they're also considered to be prokaryotic, no nucleus. So what you have is, like if I draw a bacilla here, you're going to have your DNA structure. So you'll have a chromosome. You'll have a chromosome. But unlike the eukaryotic cell, let's draw a protozoan here. It has a bounded nucleus, and its chromosome is inside that nucleus. Okay, so that's what makes it eukaryotic, is it has a bounded nucleus. It also has other, what we call organelles, arranged in there that have specific function. So you get the word organelles, meaning, like an organ. So within the cell, it's almost like, well, some of these organisms are just one cell organisms, but they're like us. They have like a body system in a way, because they have these organelles that are devoted to, respiration, energy, removal of waste. This directs the protein synthesis. The nucleus directs everything that's going on in the cell. Whereas these organisms will just have different things that do function, but they're not bounded in with a little membrane inside the cell. So that's the major difference. And of course, we know that bacteria can have flagella just like the protozoans can. Okay, so in this group, here's what we studied. Okay, so we studied bacteria, prokaryotes, and then we studied, in eukaryotes, we studied fungi, we studied protozoans, and we studied helminths. And we also mentioned as well algae, but we really didn't do much with the algae because there's not a lot of algae that cause human disease. So we were really interested in things that cause human disease: the fungi, the protozoans, the helminths, the bacteria. And then over here in this acellular group, we had viruses, and we had something called a prion, which is really more of a chemical, but it can cause infectious disease. And so we studied that as well. Now, this would share, the virus shares the fact that it doesn't have a nucleus either. It has nuclear material, but it does not have a nucleus. So in a way, there's a little bit of similarity, not much, but like you could say there's a little connection there because they have no nucleus. But then the bacteria have the connection with the eukaryotes that they have a cell, they have a cell membrane. Whereas the virus is a protein that covers over nuclear material, and that's a virus. Okay? So those were the major things, the major differences in all of those groups that we studied.
Battery's dying.
Battery's dying.
It's because it's having to focus on the whiteboard so much,
Okay.
so it's overloading.
Okay. All right, so now, Okay, so any questions on that? Any questions on that? Okay. Oh, I should have erased that. Right, so if you look on your notes there of what I mentioned for you to study, and now we're in chapter 4, and what you want to do is you want to go back, and I'm going to go over the difference between the gram-positive and the gram-negative, so I think that's really important. But you want to go back to those chapter 4 notes and review over the purpose and the parts of the bacterial cell. Go over and review, make sure that you're familiar with that. The flagella arrangement, look at that too. And I'll come back here to the structure of the cell wall in just a second. And also the 3 major morphology types, make sure you know what a cocci is, what a bacilli is, what a sporilla, and then the arrangements with the single, the diplo, the chains, and the clusters. And what was the name of the organism that's in chains?
I don't know.
The gram-positive cocci that's in chains. In chains. No, strep. Strep is chains, staph is clusters. Okay, all right, so that we want to be sure of that. And then also extremophiles, which is also in chapter 7. So basically, I think those the review games will help you there. Okay, so I want to just make a mention of the difference between the cell wall of the gram-negative and the gram-positive, okay? All right, the gram-positive organism has a large layer on the cell wall. Now, the bacteria have cell walls. Not all organisms have cell walls. In fact, most organisms don't have cell walls. That's a very, it gives the organism a rigid structure so that they're not easily lysed and not easily destroyed. So, we don't have cell walls. We have cell membranes. They also have cell membranes, but then they have that extra wall that gives them some more protection and gives them more structure. And so the gram-positive, and I'm just going to abbreviate it, GM plus, and that means gram-positive, it has this thick layer of peptidoglycan. Okay, and just, now as far as what is contained in that, that is not as important to us. It's a mesh of sugar and protein. That is not as important to us as what it means to us to be able to distinguish the organism. And then below that will be the cell membrane. Okay, so there's a layer below that, and that's the cell membrane. Right now, the gram-negative, on the other hand, GM negative, so that's the gram-negative. What it has is it has a cell membrane first, and then it has a very small layer of peptidoglycan. Okay, so this is also peptidoglycan here. I'm not going to write it again, but I'll just put PEP so you know that that's also peptidoglycan. And then it has another cell membrane. All right, so what happens when we do the gram stain, this organism, what's the color of the first stain that you do in the gram stain? What's the color of it? Violet. We use crystal violet, and so it's purple. And so what happens is that both of these organisms will take on that purple color initially, but because you have all of this thickness here, this mesh of on the gram-positive, it gets embedded in that one. Then we go across with our alcohol, and then what happens is, at this point, this one keeps it. So it keeps the violet. This one loses it out of the peptide glycan. It's just not thick enough for it to, really bind in there. And so it loses it, and then we come back with a counter stain, and so that we can see this one, and then that's why this one becomes red. Now, this one gets the counter stain too, but that doesn't do anything because it's just the color. It just kind of changes the color, but it just makes it purple. It continues to be purple. Okay?
Can you explain that red again, please? How it is red?
So the red is the fact that this one lost the color in the alcohol step.
Mm-hmm.
This one held on to its color. This one lost it.
Okay.
And so we have to counter stain with the safranin. Otherwise, this is just a little ghost to look at under the microscope. You can't really see it because it has no color. So we have to put some color on it to be able to see it. And so when you put the safranin on, it will take on the safranin, and then you can see it. And then now you know if something's a gram-negative or a gram-positive. And see, it turns out that both of these organisms, we know, staph strep, we know they cause disease. We've learned about some of the bad gram-negatives that cause disease. It doesn't mean that one's more virulent than the others, nothing like that. But it does tell us a little bit about who the organism is and what it can do, okay? So it helps us to start categorizing the organism. That's going to be the first step of identifying, okay? So that's why it's important for us. And we also know that certain antibiotics can take care of this cell wall, and that will pretty much put that organism out of commission. Some antibiotics actually can't get through that little membrane there, so they won't work on that organism. But other antibiotics can get through. So based on the cell wall is how we, something that we know about treating. So if you send out a report, so we have gram-positive cocci, then the physician has at least a list or an idea of some of the things that he or she could use to treat that organism. Okay? So any questions about that? Okay. All right. So all right, let's continue on the sheet as far as what I'd like for you to do then is, go back to your notes, and for 5, 6, 7, 8, 11, and 12, I'd like for you to look at these topics. If you have any questions, let me know. You can email me or bring them tomorrow when we have our next session. But you see that they're very defined as to what you need to study, okay? And I promise you that that's the way it's going to be. There's not, notice on chapter 5 it does talk about helminths. There's not a lot I'm going to ask you about helminths, but I would like for you to know what the two main groups were, and there were common names and there were scientific names. So go back and look at that information. We learned about a typical helminth life cycle. With the viruses, the whole idea of the obligate intracellular parasite, what that means, which basically it means that the organism has to live inside the cell of its host, and what a bacteriophage is. And if you'll notice in each of these, like 7 and like 8, chapter 8, I just put there, use the vocabulary game. So that basically means just go back and review that vocabulary game, and I'm going to pull questions from those terms that are in the game.
For which chapter, for what name?
That would be chapter 8.
Is that
It's all
some of
written.
the papers you gave us with some of the terms?
Yeah.
Or just all, is it on a computer? Because I don't have a computer at home.
Well, what you need to do is go here. You need to find out what you have in the papers, and then you can print them out here of what you don't have. So, because they're listed there, and that's the actual name that's listed on Blackboard. Okay. All right, and 11 is also. I put their the games, the names of the games. 12, and this is important for you. Chapter 12 was about the antibiotics. And we had like all the penicillins, how they affected the cell wall, and the aminoglycosides, and they did this and they did that. I am limiting what I'm going to ask from chapter 12 on the concept of selective toxicity, the whole idea of opportunistic infections due to antimicrobials, which the example is Clostridium difficile, the idea of drug resistance related primarily to the enzymes that can be produced, like the beta-lactamase or the other name penicillin. But the categories and their actions are not going to be on the final. So don't go back and do that, okay? You got a pharmacology class coming, okay? So we'll leave that for pharmacology. Again, notice in chapter 13, I just wrote the games down. That's what you need to do. You need to go back and review those games. 14 and 15, that's the immune system, and I know there's tons that we could do there. But use the games, limit yourself to inflammation, those categories of acquired immunity. Remember, we had natural, passive. You guys actually did really well on that, so just go back and review that, and the difference between the primary and the secondary immune response. So again, these topics are very targeted and focused, and if you'll pay attention to that, you should be fine. Okay? All right, that's a wrap.